Hello all, and welcome to this lecture on lymphocyte antigen receptors. As you probably remember from prior lectures from this seminar series, you can find all the figures that are included in this PowerPoint in your textbook, and if they are located in the textbook, there will be figure numbers in the upper or lower corners of the slide, as you can see, as a way for you to follow along. This can be important because some of the figures might show up with text that is kind of small, so if you want to follow along, that's how to do it. I want to start out this lecture by talking about primary objectives. These are the things that we hope to learn over the course of this lecture, so hopefully by the end we will fully understand these concepts. The first primary objective is understanding the distinctive features of lymphocyte antigen receptors and how they differ from innate immune receptors. The second primary objective is understanding the structural differences between the two types of lymphocyte antigen receptors, the T cell receptor, which is located on T cells, and the B cell receptor, which is located on B cells. Our third primary objective is understanding how these lymphocyte antigen receptors, the B cell receptors and T cell receptors, are formed during development, and they are formed by this process that is called Vdj recombination, which we will go through over the course of this lecture. The final primary objective is understanding other mechanisms that these lymphocyte antigen receptors can use to increase their specificity for antigen, and this is another diversifying mechanism. So we will talk about somatic hypermutation as well as class switch recombination. And we'll go through the mechanisms of these, but something that's important to remember about these two processes in general is that they are only happening to B cells and B cell receptors. This is not something that happens in T cells. All right, so getting started, when differentiating the innate immune receptors from adaptive immune receptors, we see over here our innate immune receptor. Here is a toll-like receptor, which is just one of the many examples of innate immune receptors. And in general, innate immune receptors will recognize a wide array of general features identified and called pathogen-associated molecular patterns, or PAMPs, as a way to recognize a wide array of different bacteria and viruses. This is a way to provide broad protective immunity that's more general as opposed to recognizing specific things that are attributed to specific types of pathogens, such as influenza virus, for example. And the other thing to remember about innate immune receptors is that they recognize a finite number of these pathogen-associated molecular patterns, and that's because they're encoded in the genome. Since these receptors are encoded, that means that they can only recognize what our genome accounts for. By contrast, when looking at our lymphocyte antigen receptors, which is our B cell receptor shown here it's shown in the secreted form, which is an antibody, and our T cell receptor shown here, something to keep in mind is that each B cell and each T cell has a unique B cell or T cell receptor for every cell. So each B cell receptor is unique and each T cell receptor is unique, which is different from the innate immune receptors. So what's important here in differentiating the adaptive immune system from the innate immune system is this specificity allows the adaptive immune system to essentially respond to any pathogen that could be incurred by the host. So there's just this wide array of diversity, and we'll explain how this diversity is generated, but just taking a moment to appreciate that the adaptive immune system is very diverse and very specific, which differs from that of the innate immune system, which you've talked about in a previous lecture. So at this point, I want to go into the structural differences between our two different lymphocyte receptors, the T cell receptor and the B cell receptor. But before we get into that, I just want to make sure that everybody is on the same page when it comes to specific terminology. Immunologists oftentimes will throw around language such as antigens, and it's important to know what that actually means. So here we can see an antigen. This is the portion that is recognized by lymphocyte antigen receptors. So here we can see two lymphocyte antigen receptors. These are B-cell derived antibodies. They're in a secreted form, which is why they're referred to as antibodies. But what's important to see here is that they are interacting with this protein molecule, and this protein molecule in this instance, because it's being recognized by a lymphocyte antigen receptor, is referred to as an antigen. To get a little bit more specific, the region that is actually in contact with the binding sites of the lymphocyte antigen receptors are referred to as epitopes, which is another term that is often used by immunologists. So just moving forward, getting familiar with those terms, and that is what they are referring to. So now moving into the comparison between our B-cell receptors and our T-cell receptors. As you can see, with B-cell receptors, there are actually two forms. There's a membrane-bound form, which will be bound to the B-cell, and during the course of an infection, when a B-cell is activated, which we'll talk about how that process happens in later lectures, but it's just important to know that as that process is happening, this B-cell receptor can change into a secreted form, shown here, and these are often referred to as antibodies. And these antibodies will actually generate and participate in the active immune response, whereas these B-cell receptors that are attached to the surface of the B-cell are mainly used prior to initiating the immune response. The major difference here is if you look at the end of the proteins, the one that's attached to the B-cell has this transmembrane domain, which keeps it anchored in the cell membrane, whereas the antibody over here is lacking that transmembrane domain, so it will be directly secreted from these activated later stage B-cells. By comparison, if we look at the T-cell receptor, this is always going to come in a membrane-bound form. So to contrast it with B-cell receptors and antibodies, while those can be secreted, the T-cell receptor will never be secreted. It will always be attached to the T-cell membrane. And you can see right here its transmembrane domain. And when we're looking at these receptors in a little bit more depth, they actually come in two different chains. So in our T-cell receptor, we have this chain referred to as the beta chain, and for reasons that we'll come to appreciate later on in this lecture, this is analogous to another chain in the B-cell receptor referred to as a heavy chain. Both of these are displayed in green, so you can see the heavy chain of the B-cell receptor here, and you can see the beta chain of the T-cell receptor here. Conversely, there's another chain to both of these receptors that is also analogous. So in our T-cell receptor, we have this alpha chain, which is displayed in yellow. And then in the B-cell receptor, this is analogous to the light chain for reasons that we will examine later in the lecture. But the important thing to remember, and an important distinction between these types of receptors, is that for the T-cell, there's only one type of alpha chain. But for the B-cell, there are two types of light chains, one referred to as the kappa light chain, and then the other is referred to as the lambda light chain. And we'll understand how this affects both B and T-cell development and their differences in subsequent lectures on B-cell development and T-cell development. But these are just important things to keep in mind moving forward. When we look at the actual structures of these heavy and light chains, we can appreciate that they are mediating different types of importances. So near the end of the T-cell receptor and the B-cell receptor, there are specific regions referred to as V, or variable regions, and as you might suspect by the name, these are the regions that are responsible for actually binding to the antigen. And they're variable because they provide different specificities. So because they're so diverse, and this is what will be different between different B and T cell receptors on different B and T cells, this is why it is referred to as the variable region. When we zoom in a little bit closer to the variable regions of B and T cell receptors, we can appreciate that there are actually two different types of regions on the variable region that's highlighted in yellow and green over on the far side of the screen. And we can appreciate when we sequence these B and T cell receptors that when we plot variability, that there are specific areas that have highly concentrated regions of variability, which are referred to as hypervariable regions or complementarity determining regions, or CDRs. For most of the remainder of the lecture, I will refer to these as CDRs, and when I'm talking about CDRs, this is what I'm referring to. In yellow here, there are these other regions that show less variability. These are referred to as the framework regions of the variable region of lymphocyte antigen receptors, and as the name suggests, they provide structural stability to the protein, and because they're providing structural stability, they don't vary as much as these hypervariable regions. So what are these regions actually doing? If we look at the 3D conformation of this protein, we see that these red sections here are actually these loops that are on the outside of the lymphocyte antigen receptors. So if we look over at our picture in the corner, you can see that the very outer edge is the red part. That is the red part here, and this is the part that will be responsible for binding to antigen, and this is why these regions are particularly diverse as compared to the yellow regions, which are providing structural integrity to the protein. And so this is just highlighted here that these red regions are on the outside where antigen binding will actually occur. In addition to the variable regions, which are on the ends of the protein that will actually bind to antigen, there are these constant regions shown here, and these are responsible for providing structural integrity to the protein. Unlike the variable regions, they remain the same, as the name constant suggests, because they're supposed to stay consistent throughout all of the receptors. So when we talk about B and T cell receptors being different and having a lot of diversity, this does not refer to the constant regions. It really only refers to the variable regions. Something else that's unique about B cells and B cell receptors that's not present in T cells and T cell receptors is they have these two arms that have the variable regions, whereas the T cell receptor, if you remember from prior pictures, does not have these two arms. And the two arms are connected by this flexible hinge region. And the way that this was studied when it was initially discovered is they found that if you digest this antibody with this nuclease called, or with this protease, called papain, this will cleave at the flexible hinge region, and you get one portion referred to as fragment crystallizable. This was just because it was easy to crystallize in biochemical experiments, but it's important terminology to know because immunologists will often refer to the FC region on antibodies. So when you hear FC region or fragment crystallizable, we're talking about this constant region that can actually mediate effector functions during an immune response. In addition to this, when we digest with papain, we get this other fragment. This is the fragment antigen binding because this is the fragment that has the variable region, which is necessary for actually binding to antigen. And as you can appreciate, there will actually be two of these fragment antigen binding regions for every one fragment crystallizable region because of these two arms. This provides us with this important concept referred to as avidity. Affinity is when a receptor will bind to an antigen. Avidity refers to when multiple sites are binding to that antigen. This increases overall affinity, and it just refers to when there are multiple sites that can interact with the same antigen. We can also see the importance of this flexible hinge region and how it could help and assist in mediating immune responses because it allows for distinct morphologies when interacting with antigen. So here we can see an angle that's 90 degrees in these hinge regions when interacting with antigen. And here we can see, this is 60 degrees, and here we can see an angle that's 90 degrees when interacting with antigen. So this just provides additional flexibility and an ability to neutralize or bind antigen in many different ways. Most of the effector functions and why this is important will come up during subsequent lectures on the immune response and how B cells help mediate that immune response, but this is a unique structural feature that is provided by antibodies. So now that we've understood the structural differences and similarities between B cell receptors and T cell receptors, I want to go into the mechanism by which lymphocyte antigen receptors are actually formed. The thing that is really unique about the adaptive immune system that's not present in the innate immune system is this diversity, like I had mentioned before. So humans are actually able to produce up to 10 to the 12 different BCRs and TCRs. So this is extremely diverse in the repertoire of B cell receptors and T cell receptors that can be produced. And this really is mediated, as mentioned before, by these CDR regions, which are located at the outermost region of the variable region. So these are the ones that are actually mediating the interaction with antigen. So you could imagine that depending on the shape of these CDR loops, that you could have different structural conformations, which would allow for accessing and binding to different types of antigens. Right here you see 4 different options of structural conformations that allow for binding antigens of different shapes. So this is the advantage provided by the adaptive immune system. But as you may have realized, this actually calls into question a problem. So humans only have about 20,000 genes. So the question is, how can humans produce 10 to the 12 T cell receptors and B cell receptors if they only have 20,000 genes? The answer to this is using somatic recombination. By generating these lymphocyte antigen receptors using combinatorial diversity, instead of using genes that are already encoded in the genome, these T cell receptors and B cell receptors can be extremely diverse. And this is mediated by a process referred to as Vdj recombination, which we will go into now. So if we look at the genomic organization of B and T cell receptors, right here we're showing the BCR heavy chain, or the B cell receptor heavy chain, we can see that it is organized into many fragments. There's variable, diversity, and joining fragments referred to as V, D, and J, which is where Vdj recombination gets its name from. And these V, D, and J segments will actually come together to form the variable region that we've been talking about present in B cell receptors and T cell receptors. Another thing that you can notice when looking at this diagram is here is the constant region. And so this is the portion of the T cell receptors and B cell receptors that remains constant. And this is also what would be referred to as the FC, or fragment crystallizable region, on the B cell receptors. To look at the genomic organization of some other portions of B cell receptors, we have our BCR kappa light chain and our BCR lambda light chain. And something that you notice here that is different from the heavy chain is that it is lacking the D segments. So the light chains will actually only consist of V or J segment, or V and J segments, but there are no D segments like are present in the heavy chain. And if we look at the organization, in T cell receptors, it's remarkably similar. So over here, we have our TCR alpha, which is analogous to the light chain, the kappa and lambda, in the B cell receptor. These consist of both V and J segments, And then over here, we have our TCR beta, which is analogous to the B cell receptor heavy chain. So it contains V, D, and J segments. Ultimately, during development of B cells and T cells, there's this process in which the B cell or T cell will pick one V, one D, and one J, bringing them together in unique combinations in order to form a heavy chain, or a TCR beta. Additionally, during development, one V and one J of the alpha chain in T cells or the light chain in B cells will be brought together to form a unique light chain or alpha chain for that particular lymphocyte. And before we get into the mechanism by which this occurs, I just want to contextualize this a little bit. As alluded to before, these lymphocytes are being formed during, the recombination is occurring during development. So for B cells, lymphocyte development happens in the bone marrow, as shown over here. In T cells, they originate in the bone marrow because this is where hematopoietic stem cells are and all the blood cells start out in the bone marrow. But this process of development and Vdj recombination primarily takes place in the thymus, shown here. So as mentioned previously, during the course of development, and right here we're looking at a B cell receptor, but this is a similar mechanism by which T cell receptors are generated, we're going to have to form a light chain and a heavy chain. The variable region, shown in red here, consists of one V, one D, and one J for the heavy chain, and one V and one J for the light chain. So we will have to pick a V and a J, or a V, D, and J to be brought together to create this variable region. And then this constant region, shown by the blue boxes, is what provides the structural integrity for these lymphocyte antigen receptors. And in humans and mice and many other organisms, but this table is showing for humans, there are many V, D, and J segments, if you remember back to the genomic organization, that this would provide many different possible combinations when picking a V, D, or J to bring together to form the variable region. So we can actually calculate theoretical diversity using these numbers. And you may notice here that these numbers will vary a little bit. Different humans might have different numbers of V, D, or J segments. And so I'm just going to use some of the numbers that are within that range for this particular calculation. So, for example, when calculating the diversity of the heavy chain, if we have 46 variable or V segments and 23 D or diversity segments and 6 J or joining segments that can be brought together in unique combination, this yields 6,348 different possible combinations for the heavy chain. But if we remember, B cells and B cell receptors consist of both a heavy chain and a light chain, so we also need to calculate the theoretical diversity of the light chain. So here, if we have 34 variable segments and 5 joining segments, for the kappa chain, this results in 170 different possible combinations. But if you remember, for B cell receptors, there are actually 2 different types of light chains, so this calculation also needs to include the possible combinations for the lambda light chain. So if we have 31 variable segments and 5 joining segments for our lambda light chain, this results in a total of 155 possible lambda combinations. So if we take both of these in total, this results in 325 potential light chain combinations. So then when forming our B cell receptor, which over in the corner you see again, which consists of both a light chain and a heavy chain, if we have 6,348 possible heavy chains and 325 possible light chains, when we multiply these together, we get about 2 million different B-cell receptor combinations. And you may notice and appreciate that this is a lower number than I had mentioned previously, with 10 to the 12th being the maximum diversity. And we will talk later on in the lecture about how there are actually additional mechanisms to help generate diversity in addition to this combinatorial diversity that is accomplished by picking up 1V, 1D, and 1J for the heavy chain, and 1V and 1J for the light chain. And here's our B-cell receptor, just to remind you of some of the potential differences between B-cell receptor formation and T-cell receptor formation. Our B-cell receptor actually has 2 light chains, which is where we got this number 325 possible light chain combinations from. But just to remember, when you're trying to calculate maximum possible diversity for T-cells, there's only a singular alpha chain. And so this will affect the calculation somewhat, but essentially the process is the same. So now that we understand what Vdj recombination actually is, let's go into the mechanism of how this is actually happening. There are several stages that I want to cover to better understand Vdj recombination. The first step will be cleavage of the DNA adjacent to these V, D, and J segments. The second step will be generating junctional diversity, which I will mention in a couple of slides how this actually is accomplished. And then the last step is bringing these V, D, and J segments together by a DNA repair mechanism referred to as non-homologous end joining, or NHEJ. So looking at step 1 of Vdj recombination, which is cleavage of the DNA, there's a specific endonuclease that's responsible for these cleavage events that occur adjacent to the V and J segments and V and DJ segments. And its recognition site are these recombination signal sequences, which you can see here. They are actually asymmetric, so one of them is called the 23 RSS, RSS being recombination signal sequence, and the other is the 12 RSS. These 12 and 23 RSSs are differentiated in this particular spacer region in the DNA that is between the actual recognition site for the protein, which is the heptamer and nonamer regions shown adjacent to the 12 and 23. So in the 12 RSS, there are 12 base pairs separating the heptamer and the nonamer. And in the 23 RSS, there are 23 base pairs that are separating the heptamer and the nonamer. And the reason that they're not listed out here is that there actually isn't a specific sequence for the 12 and 23 RSS. It can vary. But the heptamer and the nonamer need to remain relatively consistent to allow the binding of this particular protein endonuclease called RAG. Here we can see a diagram of the RAG proteins, and they will bind as a heterodimer of RAG1 and RAG2 molecules to the 12 and 23 RSS. If you notice shown here, this heterodimer will actually interact with one 23 RSS and one 12 RSS at the same time. However, it will not interact with two 12 RSSs or two 23 RSSs simultaneously. Why is that important? Well, it turns out that this actually helps to facilitate directionality of this particular Vdj reaction. So if we notice, I've highlighted the areas in which we have 23 RSSs. And you can see their recombination pair is shown on the other side. And whenever we have a 23 RSS, its recombination pair will actually have a 12. This is particularly important when considering the heavy chain shown here, and this will prevent the V to J segment joining because both of them have 23 RSSs. This asymmetrical substrate will enforce the joining of the V to the D segment shown in the middle and the J to the D segment. shown below. And so, as mentioned, this will be an important step in regulating how this Vdj recombination process actually occurs. So back to the first step, which is generating double-stranded breaks adjacent to these segments. Right here, we can see a V and a J segment, which has a 23 and a 12 RSS. The RAG protein will come and interact with one of the recombination signal sequences. In this case, it's demonstrating interaction with the 23 RSS. Then it will start to interact with the other RSS, in this case, the 12 RSS. You may notice that this is referred to as synapsis, and synapsis is when it is binding both RSSs, and this is actually necessary to bind both RSSs before initiating cleavage of the DNA. So once both recombination signal sequences, the 12 and the 23, are bound by this RAG complex, it will initiate double-stranded breaks, resulting in these covalently linked hairpin ends on the V and the J segment shown here. Now we move into step 2 and 3 after we have generated these double-stranded breaks. Step 2 being the generation of junctional diversity, and step 3 being DNA repair. So we're back with our RAG protein that has just generated double-stranded breaks. The way that this hairpin formation is shown here, it's not necessarily important how it's formed to know how it's formed, but if you're interested, it's this biochemical reaction where this side will actually be the hairpin region that you see the covalent linkage for the V and J. The hairpin actually is important when considering junctional diversity, though. So here we can see in a diagram how this is done. So the RSSs are brought together, and then when the cleavage occurs, we have these hairpin ends that are joining these nucleotides at the end of, in this case, the D and J segments. And then these hairpin ends will be bound by proteins that are part of the non-homologous end joining pathway, which is just a DNA repair pathway. Non-homologous end joining is actually used in many different processes in the cell during many different situations. This is not specific to lymphocyte development. However, we will see that certain proteins that are participating in this DNA repair are specific to Vdj recombination. These Ku proteins that will bind and initiate non-homologous end joining, though, are part of the normal non-homologous end joining pathway, and they're not specific to B and T cell development in which BCRs and TCRs are formed. So at this point, we're moving into the section about junctional diversity and how this is accomplished. There are two main mechanisms for generating junctional diversity. The first is opening the hairpins up in a way that is non-symmetrical. This is facilitated by the protein complex Artemis and DNA PK, in which they open up the hairpins, and you can see in this demonstration that they're not opening up the hairpins directly in the middle of where the hairpins were generated, but really they're skewed more to one side. And we'll see why this is important when actually looking at the nucleotides. Once again, this is a product of the DNA repair mechanisms that's an error-prone mechanism, but this is still part of regular non-homologous end joining. This is not something that's specific to Vdj recombination. Alternatively, something that is specific to Vdj recombination is the generation of non-templated nucleotides, which will also contribute to junctional diversity. This process is mediated by the enzyme TDT, which is a template-independent DNA polymerase. So, as the name suggests, it will add nucleotides to the end of these junctions after the hairpin has been opened up in a way that doesn't require a template in order to direct it. So, random incorporation of nucleotides can actually happen at this point. And so what this looks like on the nucleotide level, when we look at the palindromic nucleotides that are generated by opening up the hairpin, and as mentioned before, this is mediated by Artemis and DNA PK, when we have our hairpin and then it's opened in an asymmetric way, we have this opening that generates these ends of extra nucleotides that did not exist previously, because the ends are not generated in the middle, they're generated closer into the D and J segments, resulting in these extra nucleotides on the end that did not exist before. And then when we look at the other way of generating junctional diversity that's generated by TDT, this is the addition of non-templated nucleotides. So you can see there's just random incorporations, and this diagram actually does a pretty good job of showing that sometimes certain ends will get more nucleotides than others. So the J end was actually extended a little bit farther than the D end as it's shown in this diagram. So now that we have generated junctional diversity, which is the addition of these extra nucleotides, we can move into step 3, which is joining these ends by non-homologous end joining.